Hello, everyone. I'm Kevin Rip from Aquafix. I'm the Director of Science and Innovation here at Aquafix, aka the Bugman. And today is a very special day. We have a very interesting story to tell you about the development of our new Nitrifier Pro for nitrification and what it means to you. Helping us today is Dan McKeaton and Deborah Lee. They're both senior scientists here, and both have been studying nitrifying bacteria, their communities in wastewater, and how to culture them for 10, 12 years. So they have a tremendous expertise. So we're not only going to tell you about the Pro and what it means to you, but Dan is going to be talking about the development. And one of the things we say in our research, when we find something we're passionate about, we fail until we succeed. And Dan will talk about that, as will Deb. So Dan's going to be talking about the journey. Deborah's going to be sharing some of the data. She's going to be talking about autotrophic nitrifiers and heterotrophic nitrifiers, which a lot of people don't know anything about. And then she's going to be talking about the future, some of the future projects we're working on, and some of the future sciences that we're working on. You can see in the top, there's a downloads tab. If you want to download the spec sheet with more information on the product, you're welcome to do that. As most of you know, our laboratory is in the University of Wisconsin Research Park. We have 6 scientists who study biological communities in wastewater. We study the synergy between the different bacteria. We study the species of bacteria. We study how to influence them, how to improve them. And we do that with great passion. This is our Nitrifier Pro, if you can see it. And I'll have more information in a bit. In the past, people have bought our VitaStim nitrifiers and our VitaStim assimilators, okay? Whether it's a 1-gallon jug or 2 3.78 liter containers. That's what we've done. It's well proven in the industry. We have customers all over the world, and people really love it for restoring nitrification, getting stuff going again. What we have done over the last 2 years is figure out how to take those 2 bottles and concentrate it into this little bottle. Whereas now we ship out this little bottle with a syringe, and people will be adding milliliters of product instead of gallons. So it's easy to ship, easy to use. What are the advantages of it? Well, it restores the population of nitrifying and nitrate reducing organisms. As most of you know, they're very slow growing and sensitive to environmental changes. Deb and Dan will go into more detail on that. It guarantees full recovery in as little as 4 days. Works great in water temperatures as low as 39 degrees. And it's about 200 times more concentrate than the current VitaStim nitrifiers and ammonia assimilators, what we like to call the dynamic duo. Here is a box right here. This is what we mail to people. People no longer get 1-gallon jugs. They get this little box. And in the box, there's an ice pack, because when you receive it, it needs to be refrigerated. And there's a syringe. And right into your aeration basin, you add how many milliliters of product you need. Okay? You can order as many bottles as you want. 1 bottle if you're a smaller plant. If you're a larger plant, you might order 4 bottles. At four bottles, there's a price discount. But easy to store, real inexpensive to ship. Talk about the dose rate a little bit on this. Give you an idea. If you're operating a plant and it's 60 degrees Fahrenheit or 15.5 C, okay? If the water is above 60, you're going to feed 30 milliliters of this every day for 4 days. So if you have a 1,000,000-gallon-a-day plant, you're going to be using 2 bottles. That's all it will take. This is super concentrate culture. If it's below 60 degrees, 15.5 C, then in a 1,000,000-gallon plant, you're going to be feeding 90 mils per day. And both of those will give you very quick recovery. And like I said, Deb and Dan will be going into the data behind that. If you have a plant and you can kind of see maybe it's the middle of winter, or you got hit with toxicity and your nitrifiers are a little weak, your ammonia is starting to climb, and you can easily use half of those amounts just to boost your nitrifier population. And talking about understanding nitrification and the journey we took to get here is Dan McKeaton.
All right, thank you. So yeah, this has been a pretty long process developing these Nitrifier Pros, but before we get started talking into that development process, just a real quick review on how the nitrification process works. So nitrification is performed by two separate communities of bacteria. They're all autotrophic bacteria. They are ammonia oxidizers and nitrite oxidizers. The ammonia oxidizers are converting ammonia to nitrite and producing some acid in the process. And then the nitrite oxidizers are converting nitrite into nitrate. If you have both of these together in a community, you're able to fully convert ammonia to nitrate. But there's a number of cases where you might lose one of the populations or the other. In fact, most people are more focused on ammonia removal. So the ammonia oxidizers may seem more important, but they aren't able to function properly in the system without healthy nitrite oxidizers. And the nitrite oxidizers tend to actually get upset first in a lot of cases and recover more slowly. So those are some considerations we have in putting together a nitrifier product. So the nitrifiers have been one of our most heavily researched products. We've been working on this since 2010 as a company, and I started in 2014 and worked on it for several years when I got started. And generally speaking, it took us about a year to begin to have an effective nitrifier product after 2010. And the thing we need to keep in mind with nitrifiers in general, we've had a lot of issues with different things we've tried over the years because we have good product requirements. So is it something that can be stored? Is it something that can be shipped? Does it preserve well in different conditions? Like that sort of thing. Is it easy to produce? Unfortunately, nitrifiers, if you want to have an effective bacterial population, the nitrifiers don't make good products in many cases. Many of our attempts to make them an easier product to make have led to a nitrifier that doesn't work as well. So that's something we've had to fight with quite a bit throughout the years. So when we started research and development, and this is kind of how most research and development type projects work, first you have to figure out what do you need to do to grow nitrifiers? Where do they grow? In what type of place are they growing? We've studied nitrifiers that grow in soil. We've studied nitrifiers that grow in lakes and ponds. We've studied nitrifiers that grow in wastewater treatment. These are different communities of nitrifying bacteria, and if you put them in the wrong spot, they're not able to grow properly. Also, when growing them, we have to consider things like metabolic requirements for nitrifiers. Do you have sufficient alkalinity to neutralize the acid being produced? Is there enough carbonate present for them to use for autotrophic respiration? That kind of thing. And we also have to monitor and modify methods for culturing nitrifiers. Do we have the correct equipment? Are there ways we can improve this process so they could grow more smoothly? It took us a year to get started, like I said, but in reality, our current process took about 8 years before we got to our current phase, where we have more automation taking place and that kind of thing. And we've consistently been improving on the product performance the whole time. So we originally started with a suspended mixed culture, which we acquired from some healthy wastewater plants that had really good nitrifiers present. And what we do is we feed them the food that nitrifiers need to thrive, but tend to select away from heterotrophic bacteria that are present in most floc. So we can get a relatively consistent, relatively pure culture of nitrifying bacteria over time, and you keep reseeding and regrowing, and eventually you select for a really effective nitrifier population. The downsides of this approach is these nitrifiers always need to be kept refrigerated. They don't last for more than a couple weeks at room temperature, and in some cases, if they get really warm, less than that. And they need to be shipped with a lot of water. And you can imagine this is a challenge because if you're shipping a lot of water, it costs more to ship. It's harder to find a place to store the nitrifiers that meets the refrigeration requirements. So it's a big challenge from a product standpoint. It also takes a long time for the nitrifiers to grow. We're talking about the development of a tank of nitrifiers for product application takes several months for each one. So they're much slower to grow than a lot of other bacteria. So they're not initially a good product. You have to do a lot of work to make them work. So throughout the years, we've made a bunch of adjustments. Relatively recently, a couple of years ago, we improved the pH adjustment and we improved our aeration methods to allow the nitrifiers to grow more quickly. We reduced the room temperature so that the nitrifiers will operate better in colder temperature conditions. This was kind of interesting because we've tried growing nitrifiers down to like 33 degrees. If you make them grow in too cold an environment in the lab, they can no longer be preserved, which is a big limiter there. So we were able to find a balance between a cooler temperature so they can still work, but not so cold that we can't preserve them through refrigeration. And finally, recently we found a way where we can naturally concentrate them to make the Nitrifier Pro product. So we've tried a lot of things that did not work or kind of worked over the years, but didn't make the product good. Again, a lot of these cases, we might have made the manufacturing better, but the bacterial product would be less good. Some of the things were just kind of a disaster overall, but we started off, we did a lot of work because we knew nitrifiers grew in soil. You can imagine them on farm fields and things like that, where they apply a lot of ammonia and fertilizer. They usually apply nitrifier inhibitors because you don't want the ammonia on the field converting to nitrate because it'll wash away and the plants won't be able to pick it up. But we were seeing, good survival rates in relatively dry conditions with these bacteria. Eliminating water would have been very beneficial for the product and allow for longer-term storage. But we found none of these bacteria that were found in soil grew well in wastewater treatment type systems. So you could make a product that you can preserve on something awkward like dirt or coffee grounds or like sand, but it wouldn't necessarily work in a wastewater plant. We've tried different mixing techniques. I had a bit of a fluke, actually. I was working on experimenting with different additive powders with nitrifiers, and one of them caused the nitrifiers to form granules in a flask, which you can kind of see in the picture here, which would have been great from the standpoint of we can make the product more concentrated because these granules were very dense. They were settling out kind of like wastewater floc. But in reality, that didn't end up working well because they weren't very good nitrifiers. They ended up being very mediocre. Their ammonia uptake rates were much slower than our typical nitrifier cultures. We've tried different media. We've tried gel beads. We've tried a polymer block one company produced us one time, which we were able to preserve the nitrifiers for long periods of time in, but the block would stay in the system and not degrade, and it was such a pain to make the block that the company refused to continue making them, so we kind of had to scrap that idea. So there's a lot of things over the years that just didn't work out very well.
Okay, well, thank you, Dan, for telling everyone about our numerous failures. Until we finally have succeeded in our Nitrifier Pro, which is now this little bottle. So inside our Nitrifier Pro product, we have a lot of ammonia oxidizing bacteria, primarily Nitrosomonas and Nitrosospira. There's also nitrite oxidizing bacteria, mostly Nitrococcus and Nitrobacter, and low levels of a few other species. There's also some heterotrophs in there. Commomonas is a pretty prevalent heterotroph that we see in this product. It's a floc former that helps produce a lot of EPS to stick the floc together. It's also an aerobic denitrifier, which is very interesting because it will help you take out nitrate from your water under aerobic conditions. There's also the heterotroph hypomicrobium, which is another floc former. It turns out to be relatively important in wastewater floc formation, and that's a typical denitrifier. There's also low levels of other heterotrophic organisms. None of them are pathogenic, and none of them will hurt your system. So to test Nitrifier Pro or Vitastem Nitrifier, we do a flask test in the lab where we have ammonia at a set concentration, and we add our dose rates of products. So that green line over there is showing the ammonia decrease from our general nitrifier product, Vitastem Nitrifier. That's applied at 1 gallon per million gallons, and it took about 7 days to go from 25 parts per million ammonia to 0. Next to that middle line there is Nitrifier Pro, added at our stated dose rate, which is about 100-fold less or 200-fold less if you're comparing to both the Vitastem Nitrifier and ammonia assimilator in dynamic dual. So 200-fold less, but works pretty much about the same, but a little bit faster in this case. And then that blue line there is also Nitrifier Pro. However, that was added at the 1 gallon per million gallon dose rate, same as Vitastem nitrifiers. So you can see if you add more of the product, it decreases very quickly. However, I don't know what your budgets may allow for, but should you run into a situation where you need to decrease your ammonia faster, you can go over the dose rate, and it will improve your ammonia removals without hurting your system. So some reasons why plants lose nitrification is they're actually just losing the nitrifying bacteria themselves. So in this picture here, we see a lot of blue, dark blue there. That's actually the heterotrophs. And those small pink dots there are the little clusters of nitrite oxidizing bacteria. And the light blue is the ammonia oxidizing bacteria. So on a floc, it's a three-dimensional structure, and you're going to only see your ammonia oxidizers and nitrite oxidizers only on the outer surface of your floc. So they're a very low population compared to the dark blue, which is your heterotrophs. They're only about 4 to 6% of your total bacterial population in your wastewater plant, and they have a very, very slow growth rate. So it takes about 22 to 48 hours for an ammonia oxidizer or a nitrite oxidizer cell to double, whereas heterotrophs take only about 30 minutes to double. So they're slow growing, they're on the outside of the floc. It's not a desirable position for them to be in, but that's where they need to be to get the oxygen that they require. So because they're on the outside of the floc and they're a low population, they're highly susceptible to being damaged or broken off of the floc and washed out of the system. So one of the common ways that nitrifiers get damaged in a wastewater system is you have an influx of some sort of chemical disinfectant, say quaternary ammonia compounds, or QUAT for short. This was a test that Dan actually performed in our lab. He started off with a flask of mixed liquor from a functioning wastewater plant that was nice and healthy. So day 2, it was still pretty healthy with removing ammonia. Day 3, he decided to kill some of it with QUAT, so that's what happened, and the ammonia immediately spiked. So by day 4, you saw that increase from nearly 0 to about 6 parts per million ammonia. So he let that go for a day, and then on day 5, he added our Vitastem Nitrifier product, and you saw within 2 days the ammonia again fell down. However, in the control flask, that orange line in there is, it didn't really go down at all. The ammonia just kind of hovered until maybe something happened. And if that happened in a wastewater plant, that's pretty detrimental. So one of the reasons why you want to reseed is you've actually lost all of your nitrifiers in your plant population, and they either got washed out of the system or they were irreparably damaged. In some of the lagoon systems we see this problem happen a lot. It's just kind of the nature of a lagoon system itself. So in a lagoon, all of your floc tend to settle really quickly. That's not great because that means you're getting everything on your floc, including your nitrifiers, is getting exposed to lower levels of oxygen because they've settled. This also promotes nitrifiers to kind of break free from the floc and hover in the liquid portion because that's where their oxygens are. That's not ideal for the lagoon system because that liquid gets washed out of the lagoon and you can say goodbye to your nitrifiers. So something we've tried to do to kind of encourage nitrifiers to stick around in a lagoon is get them to grow on another medium. So in this case, that picture there is a mesh bag, and the nitrifiers do like to grow on it. They'll just stick onto the medium and hopefully remain in the water column because it's a pretty long mesh bag and not wash out of the system. And as Dan touched on, we were looking on a way to get to a point where we could ship nitrifiers without any sort of cold preservation at all, and that will be our dry nitrifiers. This product is still in development, pretty much in the early phases, so don't ask for it right now. But other than that, thank you for attending our little webinar here on introducing Nitrifier Pro.
Thank you all for attending. Anyone has any questions, reach out to us, reach out to our technical services team. They'll be glad to help you. If you ever have a wastewater mystery, reach out to us, or you get a floc, or you get something